All right, this is the review for your quiz on Monday on the DNA transcription and translation. There's the three parts to your quiz. First part is on DNA structure. It says know what a nucleotide is. So you should know that the nucleotide is the basic monomer of a nucleic acid. It's what makes up DNA and RNA. And it has the three parts. You have your sugar, your phosphate, and then your nitrogen base. Know how to number the sugar from 5' to 1' and know what bonds to what. So on this sugar, carbon 1 is always to the right of the oxygen. And then you got 2, 3, 4, and your 5th carbon is always off of the ring. And then the first carbon is always bonded to the nitrogen base. Your 3rd carbon is always bonded to the phosphate group. And your 5th carbon is always bonded to a phosphate group. Okay. And this is for deoxyribose. Ribose is a little bit different. Ribose will have an oxygen over here. Okay. Know the differences between purines and pyrimidines. A purine is a double ring, and a pyrimidine is a single ring of nitrogen base. Your purines are your ants and grandmas, right? Your adenines and guanines, and pyrimidines are your cytosines, thymines, and uracil. Know which bases are purines and which ones are pyrimidines. So we did that. Chargaff's rules for DNA is adenine equals thymine and guanine equals cytosine. So know the structure of DNA. Know that the rungs of the ladder are where the bases are. So that's where your A's and T's and C's and G's are in the middle. That's where you have the hydrogen bond, is in the rungs. The sides of the ladder is the phosphate and sugar bonds. And that's where, that's the phosphodiester bond. That's a covalent bond, super strong compared to the hydrogen bond, which is weak. So there's your hydrogen bonds, your phosphodiester bonds. Be able to label a diagram of DNA. So there'll be a diagram with a not positive if you have a word bank or not. So just be able to label it regardless. Okay, so then for DNA replication, you need to know your enzymes and what's going on. So DNA helicase is the one that's unzipping. So it's breaking the hydrogen bonds and forming the replication fork. Your DNA polymerase is adding the nucleotides. So it's adding the sugar, the phosphate, and the nitrogen base. You should know ligase is sealing up the Okazaki fragments that result on the lagging strand. And then your DNA gyrase, that is an example of the topoisomerase that helps release the strain on the molecule. And then primase produces the primer, which initiates the DNA replication. For transcription and translation, know what happens in transcription. That's when DNA is making the RNA. So that is happening in the nucleus. You have details of that with RNA polymerase. What happens in translation is taking the RNA and making the amino acid sequence, which is the protein. You do not need to know the details for the quiz, like the introns, exons, the RNA processing. I wouldn't concentrate too much on that for this quiz. Obviously, you'll have to know that for the unit exam. All right, and then you have a chart. It's going to be just like this, except you'll have different letter sequences here. So if you're not sure how to do this chart, it goes vertically. So this would be like problem 1. This is problem 2, problem 3, and problem 4. So starting with problem 1, they're giving you the amino acids. So you're working backwards. So if they're giving you the amino acid, you should be able to get the mRNA off of the code chart, which you'll be given to this on Monday, the genetic code chart. So you got to find methionine on the chart, which is right here, and get the the codon, the 3 letters for it. So it would be A, and the second letter would be U, and it's the 4th one on here, so it would be G, AUG, which you should already have known because that is the first amino acid. So that would go here on your chart. Once you have the RNA, you can get everything else, because the tRNA is the opposite letters, so UAC. The RNA is based off of the template of DNA. So up here would be TAC, and then your complementary strand would be ATG. All right, so that's what you need to be able to do. And you'll have 4 columns to do on the quiz. So this time they're giving you TAG for complementary strand. The only thing you can do with that is turn that into the original strand, which would be ATC. Once you have the DNA original template, then you can do the rest of this, because the DNA original template will give you the RNA. So that would be UAG. And then the opposite of that, or complementary I should say, is AUC. And then to get your amino acid, you always use the RNA strand, which is UAG. So if you go to your code chart, U, A, and then G is the 4th one. That is a stop codon, which I told you do not need to memorize. And so you would write stop there. All right, this time they're giving you AAU. So your DNA would be TTA. Your DNA complementary would be AAT. And if RNA is AAU, then your tRNA is UUA. And then you would look up AAU on your chart. And that would be A, so this box, and U, which is the first one, asparagine. And then you would write asparagine, or ASP is fine. The code, the genetic code that I'm giving you is actually going to have a circular one. But anyway, it has the abbreviations and the names of the amino acids here too. So, all right, last one is starting normal. So starting with the DNA strand. So CAT would be GTA, RNA would be GUA, tRNA would be CAU, and my amino acid GUA for GUA is GU. Doesn't matter what the A is. This is a wobble. Would be valine. And you would write valine or